Hey everyone, speaking with us today is Caleb Lareau, an instructor of pathology at Stanford University and Stanford Science Fellow, who is featured in the Forbes 30 Under 30 list. Dr. Lareau, would you like to tell us a bit more about your background and research?
I'd love to, and thanks for the invitation to speak with you all today. I prepared a short deck just to give some visual aids in terms of what I'll be talking about today. And please call me Caleb for the rest of the interview.
Okay.
So just to give you a little bit of background about what I've been up to and how I got here. So I grew up in a really small town in Oklahoma. The town is called Enid. So there's about 40,000 people that grew up there, and I spent the first mostly 16 years of my life there. Whenever I went to high school, I actually went to a boarding school in Oklahoma City called the Oklahoma School of Science and Math, and I was there for the last 2 years of school. So I kind of got an interest and excitement about math and science as early as high school. Really had no idea what I would be doing then. I just sort of knew math and science were my forte, and that's really where I got my start. I went to college at the University of Tulsa, which is a small liberal arts school in Tulsa, Oklahoma, and mostly studied, again, math and sciences there. So I majored in biochem and math as my two majors there. And then I did my PhD for graduate school at Harvard Medical School in Boston. So I was there for about 5 years. And then for the past 2 years, I'm doing what's called postdoctoral studies at Stanford Medicine. So the idea is that this is a little bit of extra training before I go on to be a full professor at Stanford or some other university somewhere around the United States. So this is kind of my path. It's really highlighted by all these places, and I've sort of rolled into all of these one after the other. I haven't worked in any full-time industry jobs in between. What I really spend a lot of time studying is what's called cancer immunotherapy, or you may also hear this as immuno-oncology. So this is a few different flavors of the way that we do therapies, but what today I'll be talking most about is this idea called CAR T cells. CAR T cells, the CAR stands for chimeric antigen receptor. And the basic idea here is that we're going to use DNA to essentially manipulate naturally occurring T cells in patients with cancer and turn them into really, really proficient cancer-killing machines. And so the way this works is that you do a blood draw. You pull out cells from the blood as if you would anything else, and you're going to isolate out T cells. And then what you do is you take a bit of DNA that is a specific gene that's of interest, and you put that into all of the T cells inside a dish, and then you are ultimately going to grow those cells up, and you're going to put them back in. And so the key in this process is what gene you put in the T cell that ultimately turns it into a cancer-killing agent. And once that gene is in the T cells and it's expressed, it then knows how to localize to and identify cancer cells, which it can then kill. And the way that I like to think about this is it's very similar to what happens whenever you do a vaccination, for example. So if you imagine the vaccine for COVID-19, you're training your B and T cells to recognize the spike protein as a specific thing that is associated with the SARS-CoV-2 virus. And in doing so, you have an expansion of B and T cells that specifically recognize the spike protein, and you equip your immune system to go and kill cells that then have the virus. So in an analogous sense, then once we've trained the cells, once cells display peptides from the spike protein, the CD8 T cells can know to recognize that. So in a very similar way that we have a vaccine to tell us to kill cells that have spike protein, we're going to use this thing called a CAR to kill cancer cells that have specific cancer proteins. So this is one of my favorite videos of all time, and it actually goes to show you how T cells do killing. So this is an in vitro system. So this is two sets of cells in a dish. This giant blue cell is a cancer cell, and this little pink cell is a cytotoxic CD8 cell. And what we're going to see is how exactly the T cell, once it knows what it's doing, can kill a cancer cell. So what you can see happening are there are these red granules that are getting fired into the cancer cell, and this is the mechanism by which T cells can actually go kill cancer. But you can see this cell going from this massive living cancer cell to shriveling up and like glowing, right? Super cool. And so this is essentially what we're trying to do in CAR T cell therapy is we're trying to train these little purple guys to recognize the blue guys, elicit the cytotoxic function, and then go kill cancer, which is super rad. So T cells are already in our body. right? There's lots of them, and really the question is, how does this all work? That they know how to recognize which cells to kill. So we have a variety of ways to train T cells to make this happen. Again, we tried to draw the analogy earlier that you could do this, say, a vaccine against a disease like infectious disease like COVID, but ultimately we want to target specific cancer proteins. And so you can imagine this now becomes a data science problem. If we knew what all the different cell types were in our body and what different proteins they make, you could then imagine writing an algorithm to then say which are the cells or which are the proteins that specifically express certain proteins on cancer but aren't in other cells. Because the issue is that if other healthy cells that you need, say in your brain or in your heart, also have that protein, your T cells will also kill those. So you want to find something super specific. And this is where the computational biology part comes in, is that we're essentially going to look at all proteins in all cells and then identify the most specific ones that can be used to kill cancer. And so roughly speaking, what we do is we do large amounts of making measurements and profiling both cells that come from tumors as well as from all the different cell types in the healthy body. And again, computationally, what we do is try to then identify tumor-specific targets that, again, are not on other sorts of cell types. We can plug these into a variety of precision cancer modalities. I've talked a lot about CAR T cells because it's like one of the specific ones that I focus the most on, but there's other forms of what are called bispecific antibodies or antibody-drug conjugates that also facilitate this so-called cancer precision medicine. So what do I do all day, actually? As a researcher, and that's what I am first and foremost, my job is to create new knowledge. So I'm trying to identify new proteins or new mechanisms by which cancer can be vulnerable that we didn't know before. And so that's really the job, is trying to create new knowledge, which is super satisfying because we're understanding something about the natural world in a way that we didn't previously. So we don't know the best way to kill all cancers, and I'm trying to figure that out now. I work on fairly large teams, as many as 20 to 30 people inside my lab. And we all do different things. So some of us work on creating the data, some of us will focus mostly on performing analysis, and then others, once we get hypotheses, we'll actually go and try to validate those using models like I showed you in the video of cancer cells and T cells. I'm mostly focusing on the performing analysis part of this, trying to take in lots of data and to produce new hypotheses for others to work with. So I write a lot of computer code. If you've taken a programming class, there's a lot of applicable languages out there. There's Java, among others, but the ones that I primarily use are R and Python, as well as Bash in the Unix environment. And so this is what's used to then distill all this information into finding better cancer targets. And then ultimately, I also am involved as a member of Stanford in teaching, mentoring students, as well as applying for funding to expand this work into other cancers and other interesting areas. So that's like a high-level overview of what I do. Happy to take any questions that you guys have.
Our first question for you is, when did you first realize your passion for cancer therapy?
Yeah, so I think in undergrad, I knew that I was interested in math and science, and you had always heard this adage of curing cancer is like the big thing to do, right? It's like the greatest scientific hurdle. And so I started understanding enough that I actually tried to decompose what that means. Like, what are the mechanisms of actual cancer therapy? Why aren't there better ones? Like, how would one go about actually making new ones? And so for me, that was like what happened in college. So in high school, I really just focused on, doing well in math and science classes and kind of doing conventionally the right things in terms of getting good grades, like getting good letters of recommendation. And it was really in graduate school and now at Stanford, where you're able to take a lot of that basic knowledge and then, most importantly, if you have some creativity, and actually try to find something new. So again, the original question is like, when did I first get interested? It was in college, and then it really wasn't, you're just not equipped enough to do much until you're in graduate school. And that's really when I started to produce new knowledge that I think is exciting for the field to see.
Mm-hmm. And another question that we had is, what challenges have you faced during your research?
One of the obvious ones that comes to mind is like whenever COVID happened, a lot of our work requires actually being in the lab. And so naturally, like I'm sure all of you were impacted during your training during COVID stuff, we couldn't be in the lab together to actually do experiments. And we would have to go in the mornings, sometimes in the afternoons, even evening shifts, just to be able to do experiments. So COVID's been a big one. I think the other sort of major thing that you'll run into as an academic researcher, as a professional, is trying to find the right balance between doing things that are new ideas, but not totally crazy, but also something that people can understand and are excited to support, because typically what you have to do as a researcher is convince an external agency to give you funding to do your research. So you have to find the right balance of doing something new and creative with not being too boring, but at the same time, it's like very reasonable for you to actually do it. So that's something that I think took a lot of time for me to understand, because what we're talking about now is kind of crazy in some ways, right? Like we're trying to kill cancer in brand new ways. What is that line between doing things that actually make sense versus something is too radical to actually be considered funding? You see this a lot with COVID, say, too, where people offer up new therapies or new ways of trying to deal with a disease. And so in some sense, that's what you learn as a professional scientist to do, is what actually constitutes a good idea versus a bad idea, because ultimately you're going to be reliant on others to fund your work. And that's definitely a challenge that I've had to learn and overcome.
Another question we had was, like, which moments during your research greatly changed the outcome of your work?
Yeah, I think that the major thing to internalize is that research is an incredibly slow process. You'll go from an idea to the final product, which is typically a published paper, and that can take anywhere from 2 to 3 years. So there's really never a moment where you're like, oh, this completely changes everything, because it's a long grind. But I can think back to specific ideas where it's like, oh, we should go after doing this particular experiment, or we should do this type of thing, that really impacted the outcome of what happens. But it still takes like months for that to then see it through, because it takes a while to get things together. One of the things that I think has been most transformative for me as a scientist has been realizing that it's a lot of these experiments aren't happening in isolation. So as I said, we're working with typically 30 plus member teams. And so for me, it's really been interesting to know you can actually do science with some of your best friends. Like some of my very favorite people are my colleagues and friends that I willingly would have lunch and dinner with, but then we also are working together in lab. Whenever you realize that what it entails to create new knowledge, years and years of effort and blood, sweat, and tears and effort in the lab, it makes it so much more satisfying to work with people that you know and like, and that's typically what happens. So I would say science is an incredibly social thing once you're at the level of like doing it as a professional, and so that's been a lot of fun for me to realize.
Yeah, that sounds really good. Yeah. And actually, another question that I had was, like, what inspires you to continue innovating throughout your life?
I think the biggest thing is I look at other career options, right? Like I could go any number of paths that, you know, maybe I provide like a core service or I execute some sort of known task but do it really well. And for me, none of that really sounded as exciting as creating new knowledge, which again is like the details of the job. And so for me, the motivation is really pretty simple. It's just, you know, we're humans. We don't really understand the whole world around us, and really what we're trying to do is understand why it is that things are, and try to pose experiments to basically create or falsify or otherwise establish some sort of new insights into the world around us. And so for me, like that has been, and that will continue to be the motivation. It's just like we just don't know everything yet. And as a scientist, it's incredibly empowering to set up new experiments to try to figure something out for the first time.
So one more question that I had is do you have any advice for high schoolers that want to get involved with research into human diseases, or who want to follow a career path similar to yours?
Yeah, I'd say my advice is don't feel like you need to make the big breakthrough at any particular point in your teenage years or even your 20s. It's incredibly difficult because it's a really hard job to, say, find a new mechanism for how cancer could be vulnerable. Lots of smart people with lots of energy and effort have spent lots of time trying all kinds of things to try to solve the same problem that you may be interested in, and so it's really hard. The caveat to that, and what I think my biggest piece of advice is, if you want to make inroads and you want to make new insights into human disease, is figure out like what are the new technologies that are coming online now. So in this generation, we have things like artificial intelligence, machine learning. We have new biological techniques such as droplet microfluidics, as well as things like cryo-EM. These are technologies that didn't exist a couple decades ago that give us completely new insights and completely new measurements. into either biology or into any of these other domains. And so what you'll be incredibly well positioned to do to make new knowledge whenever you progress in your career is if you become a master at a cutting edge new piece of technology. And if you know that technology extremely well, then whenever you go in and you start researching cancer, you start researching dementia, you'll have a foothold that those that have come before you don't have, and that gives you a new opportunity to find something exceptional. So I'd say don't feel rushed, like this is a long, hard game, but new knowledge will come if you stick at it. But the best way you can set yourself up for success is by mastering a new technology that didn't really exist a decade or two before. And that's sort of my recommendation for being able to advance in this space.
Thank you so much, and well, everyone, thank you so much for watching our interview with Dr. Caleb. We'll see you all next time.